Cell Transport Egg Lab by Dinah, Ike, Indy, and Buran. Summary. We'll be soaking eggs in various liquids for 24 hours. Our objective is to accurately impersonate passive transport. We'll be looking at the behavior of single cells in a new environment. In this case, the shell and size of the egg, as it will act as a big cell in the change of environment from a stable oxygen room temperature condition to a cold, harsher environment, as the eggs will be submerged in vinegar. Over time, we should see passive transport as the particles and solutes enter the cell through the membrane with the requirement of no energy being spent. The materials of the lab: 3 eggs, vinegar, corn syrup, water, timer, and 3 200 milliliter beakers. Safety materials included goggles. The lab include 5 steps. 1 being fill 3 glass cups with enough vinegar to fully submerge an egg. 2 being place 1 egg into each cup of vinegar and let it soak for 24 hours. This will allow the shell of the egg to dissolve, just leaving the membrane of the egg. Step 3 being, after 24 hours, take all 3 eggs out of the vinegar cups and replenish with new vinegar. Let the eggs sit for 2 to 3 days in a refrigerated environment. Step 4, replace the glass cups with new ones and filling each cup with a new substance, 1 with 200 milliliters of corn syrup, 1 more with distilled water, and another with a teaspoon of corn syrup, and enough distilled water to fully submerge the egg. Wait 24 hours with the eggs in a refrigerated environment. Step 5, comparing the size and mass of each of the eggs. As seen on the slide, step 2 and 3 are presented as on the image in the left shows the eggs after 24 hours being contained in vinegar and in a cold environment. As seen on the right image, me holding one of the eggs out of the vinegar, showing the dissolved shell. Step 4, submerging the eggs into 3 new beakers. 1 being distilled water, another one being corn syrup, and the third one being distilled water and a teaspoon of corn syrup. Unfortunately, we ran out of time, not being able to complete the lab, but we came to a conclusion that passive transport was accurately represented throughout the lab. We came to this conclusion because the eggs not only grew in size but mass. Once the shell was fully dissolved, the eggs started to expand within the beakers, therefore concluding the experiment.